Well, good day. Thank you for joining us. My name is Andrew Taylor, and I'm the Manager for Technical Support for Services here at XenoTech. And today I'm going to be giving a brief overview introduction of our Enzyme Induction Studies services. So first of all, just a brief overview of what this talk today is going to cover. We're going to go over what is enzyme induction and why is measuring CYP or other enzyme induction important. We'll give some just basic terminology of terms that are used in enzyme induction studies. Then go into what the regulatory guidance and expectations are. We'll talk about types of induction studies that we offer here at XenoTech, and then go into what our typical study design is, showing off the difference between our definitive study design and our screening studies. Towards the end, we'll take a look at some example results of enzyme induction, and then talk about some considerations and questions that we ask our clients up front. We'll follow this up by taking a look at some of the products that we offer for enzyme induction studies in your own lab. First, let's look at the general mechanism of enzyme induction. So enzyme induction is a receptor-mediated response to xenobiotics. So an inducing drug will enter the cell and bind to a receptor. The binding of the drug to this receptor will trigger gene transcription of the particular enzyme that's being induced. The enzyme will be converted to messenger RNA, which will bind to the ribosome and turn into protein and become an active protein. So in this example here, we're showing 4 different receptors: AHR, CAR, PXR, and PPAR. And as you can see, generally each receptor is responsible for inducing a particular gene. However, the specificity is not always the case. As you can see here between the CAR and PXR receptors, there is some crosstalk between the enzymes that they'll induce. So while generally the CAR receptor is responsible for inducing CYP2B, it can also induce the production of CYP3A and vice versa. So just keep in mind that sometimes there can be crosstalk between the different receptors. So just to illustrate how the general mechanism of induction can lead to drug-drug interactions, I've set up this little example. So in this case, the person is taking drug 1, and drug 1 is metabolized by CYP3A4. And under normal conditions, drug 1 metabolism is normal. The intended exposure to drug 1 is normal. The intended clearance of drug 1 is normal. And the efficacy of drug 1 is normal. Now, if drug 1 is also taken in conjunction with drug 2, which happens to be a CYP3A4 inducer, taking drug 2 will lead to an overexpression of the CYP3A4 enzyme. Now, with more CYP3A4 enzyme present, the normal state for drug 1 will no longer be in play. So in this case, with more CYP3A4 present, we'll have an increase in CYP3A4 enzyme. This will likely lead to an increase in CYP3A4 activity. And I say likely because the presence of mRNA does not necessarily mean that it's active enzyme. The active enzyme, increase in active enzyme will lead to increased metabolism of drug 1. And this can lead to reduced exposure to drug 1 and reduced efficacy of drug 1. Now, one of the most well-known and common examples of this in the real world is oral contraceptives and rifampin. So rifampin is a very strong CYP3A4 inducer. So when taken in conjunction with oral contraceptives, which are generally metabolized by CYP3A4 enzyme, taking the rifampin and increasing the amount of CYP3A4 will lead to increased metabolism of the oral contraceptive drugs, and therefore they'll be less effective. So just really quick, some basic terminology that's used in enzyme induction studies. I'm just going to go over EC50, Emax, and prototypical inducer. So EC50 is the concentration of the drug that gives the half maximal effect. Now, conceptually, this can be thought as it's similar to an IC50, but it's looking at the response instead of inhibition. EC50 is always a concentration, so it's presented in concentration units such as micromolar or milligrams per milliliter. Emax is the effect of the drug, the maximum effect of a drug at concentration C. So Emax is represented as a fold change compared to a vehicle control. So for Emax values, it'll be something like 2-fold, 3-fold, etc. Lastly, a term that's used a lot is prototypical inducer. And a prototypical inducer is a compound that's known to induce a particular enzyme. And we use these as a positive control. Some examples of this are, as I just mentioned, rifampin is a prototypical inducer of CYP3A4, as is phenobarbital. Okay, meeting the regulatory expectations. With the current introduction of the new 2020 guidance from the FDA, we're seeing a convergence on the guidance requirements between the FDA and EMA. And this makes things a little bit easier for us in industry. So for the test systems that need to be tested when looking at enzyme induction, both the FDA and EMA recommend using cryopreserved, characterized, or fresh human hepatocytes. Now, other systems can also be considered if they complement this and there's a need for them. Both regulatory agencies suggest that a minimum of 3 donors are looked at. And here at Sekisui XenoTech, our main test system of choice are cryopreserved human hepatocytes, which have been pre-characterized using mild to strong clinical CYP3A4 inducers. However, upon request, we can also use fresh human hepatocytes. And again, we use 3 hepatocytes from 3 donors. The TA concentrations to be tested in the studies, according to the FDA, needs to be sufficient to reach Emax, but it's not based on the Cmax at steady state total unbound concentration. While the EMA recommends looking at 50 times the Cmax steady state unbound, or if the drug is orally dosed, the concentrations, the maximum concentration should be 1/10 of the dose in 250 milliliters. Again, at Sekisui XenoTech, our standard study design for the maximum concentration is to look at 1/10 the dose in 250 milliliters, or 50 times the Cmax steady state total unbound. And of course, the maximum concentration that'll be tested could potentially be limited by solubility or cytotoxicity of the test system. The CYP enzymes that are recommended to be looked at are the same for both regulatory agencies, and that's CYP1A2, 2B6, and 3A4. And both regulatory agencies recommend using CYP-specific positive controls. Now, the positive controls between the 2 regulatory agencies are the same for all the CYP enzymes, except for CYP3A4, where the FDA recommends using phenobarbital as the prototypical inducer, while the EMA recommends looking at CITCO. Now, here at Sekisui XenoTech, we use phenobarbital as our positive control for CYP3A4. Now, some might think that the reason we do this is because we're located in America and a majority of our clients are American-based and submitting to the FDA. However, that's not the case. We've chosen to use phenobarbital over CITCO because phenobarbital gives a much more reliable response rate. So it's easier to compare back to historical data. The endpoint measurement requirements from the regulatory guidance agencies are to look at messenger RNA. And for our induction study designs, we can look at mRNA and or the activity of the enzyme. A positive induction response, so this is the induction response to the positive controls, is not specified by the FDA, but the EMA requires that a 6-fold increase in messenger RNA from the prototypical inducer be seen. Our standard is to look at a 6-fold increase in messenger RNA. The concentration of the test article in the medium should be checked according to both guidances, and our study design does include a spent media collection on the last day of the treatment, and this is taken over 4 different time points, and the amount of test article in the media is looked at using a specific LCMS method, or the spent media analysis samples can be stored for future analysis. Toxicity assays are required by the EMA, and it is our recommendation to include cytotoxicity assays, either using lactate dehydrogenase or the reduction of resazurin. Individual versus average cultures. Both regulatory agencies recommend looking at individual cultures, so not pooled hepatocytes, but individual cultures. And if induction is seen in any one of those cultures, it's considered a positive induction response. This is just looking at a worst case scenario for the drug. Various methods can be used for the data interpretation according to the regulatory agencies, including basic models, mechanistic static models, or dynamic models. And GLP compliance is not required by any of the regulatory agencies for in vitro induction studies. However, they are available upon request. And including GLP compliance into your study will also require including a dose solution analysis. So there are two main types of induction studies that we conduct here at Sekisui: ex vivo and in vitro studies. Now, for ex vivo studies, the investigations of induction are conducted in laboratory animals, usually rat, mouse, dog, or monkey. These are typically done following toxicology studies by the sponsor as a GLP multi-site study. The animals are generally dosed by the sponsor, and then the liver samples are sent to us at XenoTech for analysis. And the samples are tested for either looking at the microsomes for enzyme activity or RNA from the individual animals, and these are isolated from the liver tissue that's sent to us. Now, the majority of our studies, induction studies, are in vitro studies. For in vitro studies, enzyme induction is examined in cryopreserved characterized hepatocytes. The cultured hepatocytes are treated with test articles for 3 days. Only small amounts of the test article are required since we're doing these in microplates. Hepatocytes from rat, dog, mouse, monkey, or human can be used. Again, in vitro studies, we can either look at microsomal activity of the enzymes or RNA from the treated hepatocytes. So for our in vitro enzyme induction studies, there are two different study types that can be chosen from. These are our definitive study design and our medium throughput study design. Our definitive enzyme induction study design is designed to cover the guidance recommendations from all the regulatory agencies. This design typically looks at mRNA fold change for CYP1A2, 2B6, and 3A4 using reverse transcription PCR. 3 lots of hepatocytes, so 3 individual donors are looked at over a 72-hour treatment period, and these are done in true triplicate biological replicates. 6 to 8 TA concentrations are used. That way, if any induction is observed, an EC50 or Emax can be calculated. The study design includes a vehicle control, a negative control for induction, and multiple positive controls for each CYP enzyme, and the multiple positive controls are done at 1 concentration. This also includes spent media analysis at multiple time points on the last day of the experiment, and it includes a full FDA submission ready report. Additionally, if induction of CYP3A4 is observed, we can follow up looking at induction of CYP2C8, 2C9, and 2C19, either again using activity or mRNA. We can also include GLP dose solution analysis, and pre-induction study cytotoxicity assessment can also be included. Now, this is contrasted by our medium throughput study design, and the medium throughput study is more of a screening type study. This study design also looks at RNA fold change for CYP1A2, 2B6, and 3A4. In our medium throughput study design, we only use 1 lot of hepatocytes, and they're only treated for a 24-hour period. Only 3 concentrations of test article are included. And while we do these in triplicate, they're pooled measurement, so they're not true biological replicates. A positive control is included. With our medium study design, we require that the test article be shipped to us either already in a solution or as a pre-weighed aliquot. The medium throughput study design does include lactate dehydrogenase to assess cytotoxicity. And instead of a full FDA submission report, it only includes a tabular data summary. Okay, just to take a quick look at some example induction data. Now, just to get you oriented to both of these plots, they're both set up the same way. The y-axis is the different treatments, including the vehicle control, negative control, and positive control, along with increasing concentrations of the test article. And the x-axis is the fold change in mRNA. Now, as you can see here in this first graph, this is a very good example of a clean, concentration-dependent positive response. As the concentration of the drug increases, we also get a very strong fold change that also increases with the increasing concentration. And just over here is the negative response to contrast that. Again, on the y-axis is our different treatment types from the vehicle control, increasing concentrations of the drug, and then negative positive control. And as you can see, as concentrations of the drug increase in this example, we don't see any increase in the fold change. However, you see that the positive control has a very strong fold change, illustrating that the test system was functioning properly. It just happens in this case that this drug was not an inducer of this enzyme. Now, if we take that same data and just plot it in a slightly different way, we're able to look at EC50 and Emax. So again, these three panels are the data from three different donors. The y-axis is the effect, so it's the fold change, and the x-axis is the concentration of drug. So here we're looking at the fold change with respect to the concentration of drug. And as you can see, as the concentration of drug increases, we see an increase in the fold change or increase in induction of the particular enzyme. So by plotting the data this way and then fitting a line to it, we're able to estimate the Emax and EC50. So induction data interpretation. What do we do once we get this data? Well, we determine the fold change of the enzyme compared to the vehicle control. And we looked, did the test article cause a fold change in RNA activity levels greater than twofold? And did the test article cause a response in mRNA and activity levels that's greater than 20% of the positive control? And if both of these are the case, then according to the guidance, it's considered an inducer of that enzyme. Another thing we like to look for is, were these increases observed concentration dependent? If the increases are observed, we determine the EC50 and Emax values, and additionally, R3 values can be calculated according to the FDA 2020 guidance to determine if that response in induction is going to be clinically significant. Alternatively, we can compare the test article response to a range of weak to strong inducers and develop a relative induction score, but this is done for CYP3A4 only. Some additional considerations to take into account are, was any cytotoxicity observed? Does the mRNA response match the response in activity? As I mentioned before, an increase in RNA does not necessarily always correlate with an increase in activity. And should a mechanistic prediction model be used? Okay, now I'd like to go over some considerations and questions that we like to get from our sponsors when determining the design of their in vitro enzyme induction studies. Now, one of the first things that we like to get information on is information on the test article. So are there any specific considerations on the test article that we need to know? These include things like solubility, any strong non-specific binding of the compound, stability information of the compound, the molecular weight, what type of molecule is this? Is it a small molecule? Is it an oligonucleotide? Is it some sort of large biologic? Things like that really help in designing the experiment. Also, it's really good to know what questions are you as the sponsor trying to answer. Are you just checking the FDA boxes in a human study and just getting that done? Or are you doing this in different types of animals to determine a tox species? So any type of information like this is always really important to know. Another important thing to know is, do you plan on submitting to the FDA, the EMA, or both? While our definitive study design is designed to meet the requirements of all three regulatory agencies, FDA, EMA, and PMDA, it's always good to know up front where you plan to do with these data. For example, if this data is going to be used for additional modeling, we might do some things like throw in some additional positive controls for an induction to make that data set slightly more robust. Also, some of our clients might also want to include EC50s of the prototypical inducers that are used in the experimental design. So it's just good to have that information up front. Lastly, it's always really helpful to know, if it is known, the Cmax unbound of the drug. What are the physiologically relevant concentrations that the drug is going to be dosed at? And also, is there any plasma protein binding? When conducting ex vivo enzyme induction studies, we also have a list of considerations and questions that we like to ask our sponsors, and these include what species are we looking at, the number of animals per sex, and are these going to be pooled or individual samples, the number of treatment groups, is this study being conducted GLP or non-GLP, is it going to be a multi-site work plan, or are the samples just going to be sent to us as a standalone study? We need to know specifically what CYP enzymes or other enzymes such as UGT are going to be analyzed for induction. And again, what specific questions are you trying to answer with the study? We also need to know what endpoints are going to be required. Are we just looking at mRNA or activity? If looking at CYPs, we like to use CYP-specific probe substrates. And if you're wondering if we have a preferred method of preparing the livers for our ex vivo experiments, we do, and we're happy to share a protocol with you. Lastly, at Sekisui XenoTech, we offer a number of products that can be used for CYP or UGT induction studies if you wish to conduct them in your own lab. These include cryopreserved attaching primary hepatocytes from individual donors, and these have all been characterized. We have pooled primary human donors for screening studies, and multiple small animal species are also available. Along with our hepatocytes, we have a number of support reagents to be used for culturing these cells. And we also offer a number of immortalized hepatocytes that are sometimes used in induction studies. And again, we have support reagents for all these immortalized hepatocytes as well. Additionally, we have treated animal liver subcellular fractions that can be used as positive controls for induction studies in your own lab. And a list of these are shown here. And on that note, I'd like to thank you very much for taking the time to watch this video today. If you have any questions or are interested in setting up your own enzyme induction study, please get in touch with us through the Contact Us tab on our website. And you can also look at our products pages to find any of the products that I just mentioned if you wish to conduct these studies in your own lab. I'd also like to point out a video by our Vice President of Scientific Operations, Dr. Joanna Barbara, who has a very good, more in-depth presentation on enzyme induction, and you can find that here on our website as well. Thank you very much and have a great day.